Thank you. And yeah, thanks for inviting me to give this presentation. I'm excited to share this work with the biocars community. I see a lot of familiar faces on the call here and also a number of people who I haven't met before. So it's always fun to share work with new people. And yeah, as Irina mentioned today, I'm going to talk about temperature jump X-ray crystallography experiments. And before I get started, I'm just going to thank all the many people that contributed to the work that I'm going to talk about today. So the folks from my lab specifically who contributed are listed here. And I want to especially highlight Alex Wolf, who has been working with me on these temperature jump projects essentially since the beginning and has really contributed to almost everything that I'm going to talk about today. First, I'm going to describe some work that we published last year. And the folks who contributed to that work are listed on the publication here. And then in the second half of the talk, I'm going to transition and talk about some newer work that hasn't been published, but which also represents a big collaboration involving many people, most of which are listed here on the right-hand side of the slide. And of course, I want to thank the funding agencies. So I think an idea that's going to be familiar to most of the people on this call, but that I want to just begin with to kind of frame what I'm going to describe is the idea that protein dynamics are complicated. So proteins have lots of atoms, and of course those atoms move, and those motions involve a hierarchy of different length scales and time scales. So with this figure, I'm trying to depict that all of the atoms right within this protein are undergoing motions that include sort of fast time scale vibrations, things that happen on sort of the picosecond to nanosecond time scales down in the basins of these wells in this landscape. And there are also slower motions, things like the nanosecond time scale exchange of some rotamers, and for example, millisecond time scale motions that involves for perhaps slower, more collective transitions. So building up this picture, right, of sort of a hierarchy of motions that involve both different length scales and time scales. These types of motions that I show on the slide here are also coupled to larger scale motions like this domain closure, this clamshell motion that we see in the enzyme shown on this slide, which is lysozyme. And lysozyme will be a key player in the results that I describe later on in my talk. So really, I want to, just kind of summarize the idea that proteins have complex motions, and those motions are difficult to study. And one of the sort of tried and true methods that we use for studying protein structure, of course, is X-ray crystallography. And one of the benefits of X-ray crystallography is that it's an ensemble method. So we're measuring billions of molecules at the same time. And if those billions of molecules are in different conformations, then what we see over here on the left is an ensemble averaged electron density map that shows us multiple different conformations that are accessed by the protein. And so this is nice because we can see this conformational space. The problem with crystallography is also that it's an ensemble technique. And so this spatiotemporal averaging that can be an advantage in terms of identifying alternative conformations also results in the loss of information about dynamics. And I've tried to depict that idea over here on the right side of this slide. So in the ensemble averaged electron density that you see on the left for these two amino acids, this serine and this phenylalanine, you can imagine those conformations, those individual conformations for each amino acid exchanging in different ways, right? They might be completely coupled to each other, and that would give sort of an energy landscape that looked like this, where there's a single kinetic step that involves the transition of both of those amino acids at the same time, both of those rotamers at the same time. Or you could envision a situation where those motions are decoupled from each other. And so you have a more complex conformational landscape and multiple motions that result in the transition from the red state to the blue state. So this is a challenge, and this is a familiar challenge to biophysicists, right? Because we've been studying things that are in motion for many decades, right? Going all the way back to these very early high-speed imaging experiments that were done by Edward Muybridge in 1872, right? Edward Muybridge built the world's first high-speed camera. Back in the 1800s, if you were to use a normal camera to try to capture a picture of a horse at full gait, you would see something like this, where all of the horse's limbs would be blurred out because of their motion is faster than the exposure time of the camera. But in 1872, Edward Muybridge built the world's first high-speed camera and captured these very, very famous pictures of the horse at full gait, which those of us who do time-resolved imaging are, of course, very familiar with these images because they're sort of a classic example of the problem that we're trying to solve here. But in the case of Muybridge, he had it a little bit easier than we do because Muybridge could take a picture of a single horse. In the case of crystallography in particular, we have, as I said before, right, many billions of molecules in our crystal, and we're imaging all of them simultaneously at the same time and looking at the ensemble average. So we, not only do we have to take rapid images, but we also have to synchronize our samples. And this is done, of course, in time-resolved crystallography, which essentially acts as a high-speed camera for molecular motion. And so, again, an idea that's probably familiar to most of us on this call is that the way these experiments are done is that we apply an instantaneous perturbation that takes our system out of equilibrium. And then we're going to use short, bright X-ray pulses, like the ones that are available at biocars, or in the case of the data that I'm going to show today, available at X-ray free electron laser sources. And those short, bright X-ray pulses act as a high-speed camera that allow us to capture images of the molecules undergoing transitions from one conformational state to another as they relax to a new equilibrium. So traditionally, or maybe I shouldn't say traditionally, but time-resolved crystallography was really pioneered using light-activated proteins. So there have been really beautiful experiments on a variety of different photoactive molecules, including things like bacterial photoreceptors, other types of photoreceptors like the rhodopsins that are found in the eye, heme proteins, which interact with light to release carbon monoxide from the heme center, and also light harvesting proteins, such as the ones in photosynthesis. And this list is probably not inclusive as well. I'm simply trying to make the point that these experiments were really, time-resolved crystallography was really pioneered on light-activated proteins. And there's an important technical reason for this, which is that it's fairly straightforward to get them to undergo synchronous conformational changes. So you keep the crystals in the dark, and then when you're ready to initiate your conformational change, you shoot them with a pulsed laser that's tuned to whatever wavelength the chromophore absorbs, and then you can activate your conformational change of interest. So light-activated systems provide a really nice means by which to initiate these experiments. The problem is that if we consider the entire universe of protein molecules, the fraction of proteins that have some sort of appreciable interaction with light, some sort of functional interaction with light, is quite small. So I made this little pie chart here by searching through SwissProt, which is a manually curated database of proteins. And I searched for terms like photoactive and photoreceptor. And what I came up with is about 0.25% of all proteins are associated with some type of photoactivity. And for me personally, throughout my career, I've always really been interested in enzymes. And there are some photoenzymes out there, but the majority of enzymes fall into this large blue slice of the pie. So I was always really enamored with time-resolved crystallography because the experiments were so beautiful and so informative. But I always felt like they were a little bit out of reach for the molecules that I wanted to study at the time. So when I started my postdoc in 2014, we thought that we could potentially address this problem using temperature. So it's been known for decades now, and the data that I'm showing here on the left is from a 1992 paper from Greg Petsko's group, that you can alter the distribution of conformational states in crystallized proteins using temperature. So in this old paper from Greg Petsko's group, they showed that if you just calculate the average Debye-Waller factor, average B factor of the atoms in your protein crystal as a function of temperature, there's a dynamic transition around 200 Kelvin, and then above that point, there's a really essentially linear relationship between the disorder as measured by the B factor here and the temperature. And when I started my postdoc in 2014, I was involved in this study that I show on the right, where we did essentially the same experiment that Greg Petsko's group did over here with RNase using cyclophilin A. And we looked in atomic detail at how the conformations change as a function of temperature through this, this regime from 200 Kelvin up to above 300. And what was really striking, and you can see it most clearly if you focus on the phenylalanine here in this network of amino acids that are undergoing this conformational change. As we raise the temperature and get above this dynamic transition, you start to see this green blob of positive difference density that gets stronger and stronger as we go to higher and higher temperature. And that essentially corresponds to an increasing population of this alternative conformation at higher energy. And so this, this work that was published in 2015 really kind of highlighted to us that we can use temperature to shift the conformational ensemble in a way that is observable with X-ray crystallography. Whoa, I'm writing all of a sudden.
Sorry, that was me. That was on accident.
Okay.
I'll delete it.
So yeah, so we realized that we can observe this on the atomic scale as we increase the temperature in our crystal structure. So that suggested to us that maybe we could use temperature in a time-resolved experiment. And of course, this is not a new idea. Temperature jump is an idea that's been around in physical chemistry for many decades and has been used not only to study chemical kinetics, but also to look at things like protein folding, for example. And the idea behind an infrared temperature jump is that you can use the photochemistry of your solvent rather than photochemistry of the protein, right? So if we irradiate water at, in our case, 1440 nanometers, then we excite this OH stretching overtone, and that kinetic energy, that increased vibrational energy gets dissipated into kinetic energy, which results in a rapid heating of the sample, shown in this schematic on the right. And that heating is faster than the changes that we're interested in observing, effectively instantaneous. And then conformational changes of the protein are slower. And so then we can sample this, this evolution of the system, right, as a function of time using more fast X-ray pulses from either biocars or from our free electrons. And temperature jump is a really nice method for studying proteins because when we have proteins, we always have water, right? Proteins live in water. And so if you're studying proteins, then there's water around that you can pump and heat that up. So we thought that this would be a really nice way of a universal way of perturbing protein conformational changes in a time-resolved experiment. And as Irina already alluded to, our first successful attempt at doing this was actually using solution scattering at biocars in collaboration with Philip Anfinrud and his group. And I'm not going to describe this work today, but I did just want to point it out because it's sort of my connection to the biocars community here. The work that I'm going to describe today, which was done in a free electron laser, is sort of really the evolution of our early work at biocars. So I'm grateful for this big collaboration, again, involving Philip and his group, as well as the folks at biocars. And I also want to point out that in addition to our work, which I've cited here, there's also really nice work from Lin Chen's group, from Philip's group, not including us, as well as more recently from Rory Henderson's group, using T-jump to study conformational changes using the solution scattering And we heard, I think it was relatively recently actually, about this work from the Henderson group. I'm very happy to see more and more people doing these temperature jump experiments. They're really powerful. So with that, I'm gonna start, really kind of the meat of what I want to discuss today, which is our temperature jump crystallography experiments. So I kind of framed the problem, and I mentioned that the solution that we came up with was that we're going to use infrared temperature jump as a perturbation method in time-resolved crystallography to make this experiment general to really any protein of interest. So this is the schematic showing how this experiment works. And our first successful T-jump crystallography experiment was performed at the SACLA free electron laser in Japan with Eriko Nongo's group and Sowawada's group. But prior to that, we had done a number of kind of pilot experiments at LCLS. So it really took the effort of a lot of folks to eventually get this experiment to work properly. And the way we settled on doing it was using a microfluidic serial crystallography approach. So for anyone who's unfamiliar, the way this works is that we first grow a slurry of small microcrystals. So the crystals that you're seeing in this image here are on the order of 10 microns in each dimension. And we load those microcrystals into a microfluidic injector that essentially streams them into this interaction region where they get pumped with a nanosecond IR laser that's tuned to 1440 nanometers, which excites, as I mentioned, the OH stretching mode of our water molecules. And then in this particular case, we are probing our sample at some defined time delay following that IR pulse using a X-ray pulse from a free electron laser that in the experiments that I'm going to describe today is anywhere on the order of 10 to 30 femtoseconds. So very, very fast experiments. Okay, so this is the overall schematic describing how the experiment works. And we applied this approach in our pilot experiments to lysozyme, which of course is every crystallographer's favorite protein to do method development on because it crystallizes so easily, it diffracts so well, and you can dump it out of a bottle and crystallize it. But fortunately for us, it also has some relevant dynamics. So earlier in my talk, I showed the animation of this clamshell motion in a protein, and it turned out that was lysozyme. And the way that this enzyme functions, the active site runs through this cleft in the center of the enzyme. And when the substrate binds, the enzyme sort of clamps down. That's that clamshell motion that we saw before. And that clamping down on the substrate then positions all of the catalytic machinery in the correct place for the catalytic step of the reaction. So it's a nice enzyme for crystallography related reasons, and it also has some well-established dynamics that we can look for in our initial attempt to do temperature jump experiments. So this was the same. This is a picture of what the actual experimental configuration looked like at the SACLA X-ray free electron laser. So coming from the left of this slide, we have the X-ray optics and the beam pipe that are delivering our X-ray pulses. In the center of the photo here, this is the sample chamber, where at the top we have our device for microfluidic sample delivery, which I'll show in more detail in just a minute. This other black pipe that you see here, which is kind of going into the direction of your computer screen right now and then wrapping around the back of the sample chamber, carries the IR laser beam and the optics that are required to deliver it to the sample chamber. Over here, we have an IR camera, which is important, of course, for monitoring the alignment of the IR pump laser. And then behind the sample in a transmission geometry, we have our X-ray detector. As I transition to this next image, what we're going to do is rotate 180 degrees around the experimental setup here, and then zoom in. So that's what you're seeing here. And now you can see in more detail what goes on in the actual interaction region here. So this nozzle that you see is the microfluidic nozzle that delivers crystal sample. Now our X-ray beam pipe is on the right, and the X-ray beam is coming from right to left to arrive at our sample. And you can also see here the final focusing optic that delivers the ion. Then, of course, over here now on the left, we have our X-ray. I want to also mention a quick word about sample prep, which is that we take our microcrystal slurry, and before we load it into this microfluidic device, we embed the crystals in 15% cellulose. And 15% cellulose has the consistency of like a thick jello. And that facilitates a really smooth injection of the sample into the interaction region. So what you're seeing over here is a video showing the sample streaming out of the microfluidic nozzle. And what I hope you can appreciate is that the flow of the sample is really consistent and uniform. And that's really helpful for alignment of both the pump laser and the free electron laser, and getting good spatial overlap so that this experiment works well.
Michael, might you comment on the dimensions of that ribbon that's going through and the relative size of the crystals, because that affects background issues?
Yes, absolutely. So this, the diameter, the inner diameter of this nozzle is about 75 microns. So it's substantially wider than an individual crystal. And you are right that there's a fair amount of background. And in a moment, I'll show a diffraction image, and you'll notice that there is quite a bit of background. And in the case of lysozyme, we're fortunate because the crystals diffract so strongly. But I think you make a good point that in the limit of poorly diffracting crystals, this could potentially be problematic.
Thank you for that clarification.
Sure. I also really like to show this image here. So for anyone who's unfamiliar, the free electron laser pulses, one of the reasons that we have to use this serial crystallography approach is because the individual pulses are so powerful that they essentially destroy the sample immediately. Fortunately, the diffraction patterns can be collected within tens of femtoseconds before the crystals explode, but you don't get another shot. We call this principle diffraction before destruction. So that what that means effectively is that we need to keep refreshing the sample in the interaction region. And I like to show this picture not only to highlight how powerful the X-ray beam is in these experiments, but also to show how uniform the flow of sample is out of that microfluidic nozzle. So what you're looking at here is this cellulose material. As it comes out of that injector, it sort of dries up and forms almost like a string. And so we pulled the string out of the sample collector after the experiment was over and looked at it under a microscope. And you can see that there are all of these holes in the cellulose that have been drilled by the X-ray beam. And what's really striking is how almost perfectly spaced each of those holes is, right? And so what that tells us is that we have a really nice, consistent linear flow rate of our sample during the experiment. And as I alluded to a few minutes ago, we are able to collect some pretty nice diffraction patterns. There are some weird detector artifacts here that have to do with the CCB that is in this SACLA detector. But otherwise, the diffraction patterns are quite strong. The diffraction data can be processed to about 1.6 angstroms. And to Philip's point, there is a fair amount of background, but in the case of lysozyme, as I mentioned, we have very strong diffraction. So we're able to process those data using the standard approaches for indexing and merging serial diffraction. I'm also going to say a few words about our data collection scheme in these temperature jump crystallography experiments. So the infrared lasers that we tend to use for these experiments almost universally operate at 10 hertz. And the detector that we like is this rayonics detector. It's similar to the rayonics detector at Biocars, and that operates at 30 hertz. So what this allows us to do is basically collect two interleaved images where we have no T-jump. So for every temperature jump measurement that we collect, we get two what we call dark images where we measure X-ray diffraction, but in the absence of any temperature jump. And that's very useful because then after we process the diffraction data and extract the structure factors, then we can make some useful difference maps. So for example, we can calculate what we call the experimental maps, where we take a difference between the T-jump images, referred to as the light images here, and the dark images, in this case, the dark 2 images. And we can also make a control map where we subtract the dark structure factors, dark 1 and dark 2, from one another. And if we really have no T-jump, then there should be no signal in these maps, or any signal in these maps should represent the overall noise level in our experiment. So at first, we weren't too thrilled that we couldn't collect T-jump data at 30 hertz, but we came to realize that this is actually a blessing because these interleaved dark measurements are really important. And this also reflects the way that we've collected solution scattering data at Biocars. I think this is just good practice in general for the time result experiment, which is something that we learned from Philip and the Biocars. Okay, so I'm not going to go into the details because I don't really have time, but we can use the solution scattering in the background of our X-ray diffraction to calculate to first of all determine whether or not we've introduced a T-jump, and in favorable cases to calculate how large that T-jump is. This is something that's also been done by us and others in our solution scattering paper. So if you're interested to know how that works, you can look at those references. And I really want to move on and get to talking about how the protein responds to temperature jump in these experiments. So the first thing that we did was to just look at some very simple parameters. First, we looked at the unit cell dimensions of our crystals in the presence and absence of temperature jump. And what you'll notice, so here we're plotting the A and B axis lengths. So lysozyme, we use tetragonal lysozyme for these experiments. So A and B are by definition the same length. So we're plotting the AB axis length as a function of the pump probe time delay. In the experiments that I'm going to describe today with lysozyme, we have very limited sampling of the time domain. We were only able to measure 3 pump probe time delays. Those were 20 nanoseconds, 20 microseconds, and 200 microseconds. And so you're looking at the AB axis length as a function of pump probe time delay. And what you'll see is that there's a very small but real thermal expansion that we see when we apply the pump laser. So again, this is a small thermal expansion, but in these serial crystallography experiments, our data set consists of tens of thousands of crystals. And so we get very small error bars on these unit cell axis measurements. And also what's really interesting is if you plot the AB axis lengths for the corresponding interleaved dark images from the same crystal batch, you don't see a thermal expansion. Those unit cell axis lengths are much more similar to what we refer to here as the true dark, which corresponds to a data collection run where we had the IR laser completely turned off. So there were no T-jump measurements at all. Also, with the reduced diffraction data, we can do some very simple structure refinements. And those refinements produce not only coordinates, but of course, B factors. So we can look at the average atomic B factor, again, as a function of pump probe time delay. And we see that for the experimental T-jump measurements, but not for the controls, we see a rapid increase in the B factors at 20 nanoseconds. And that increase in B factors seems to dissipate as a function of pump probe time delay. So that was nice. And so, this panel on the right here comes, as I mentioned, from refinements of our structures at each of these 3 pump probe time delays. So the first thing we did was sort of the simplest thing, which was we took all of those refined structures and we compared them. And we asked whether or not we could see any obvious differences. And unfortunately, and perhaps unsurprisingly to other folks who do similar types of experiments, we didn't see any major differences in the average refined coordinates. So at first we were disappointed in this, but then we realized that really what's going on here is actually just that the excited state populations are really small. And of course, when you refine a single conformation in a protein crystal structure, you're always going to end up refining sort of whatever is the most dominant thing there, right? That's kind of what's going to dominate the average. And so we started looking a lot more carefully at our difference electron density maps. And 2 of those are shown here for the 20 microsecond time delay and the 200 microsecond time delay. And we're looking kind of zoomed out here over the entire lysozyme molecule. And I will note that we use this structure factor, this different structure factor weighting scheme that's been described as far back as the early 90s and is used relatively often in the generation of difference maps from time resolved data, and applying this weighting scheme is actually really helpful for removing some noise from these difference maps. And so the reason that I'm showing the image that I show here is just so that everyone can kind of appreciate from looking globally here that the character of the difference density changes as we go from 20 microseconds to 200 microseconds. So you'll notice that at 200, there's substantially more difference density than what we see at 20 microseconds. And it's really concentrated in this lobe of the enzyme here, which is right above the active site that runs through this channel. So it was encouraging to see that. And so we dug in a little bit deeper. And the first question that we wanted to ask is, is this meaningful difference density or is this noise? So what I'm showing on this slide is a comparison of our control measurements with our experimental measurements. So here the columns represent the different pump probe time delays in our experiment, and the rows represent either the control on top or the experimental maps on the bottom. And just to remind everybody, in the case of the control, that's where we're subtracting the 2 laser off measurements. And in the experiment, we're subtracting the laser off from the laser on T-jump image. And you can see there's a very clear difference. The control measurements show almost no signal, whereas the actual T-jump images, particularly at shorter pump probe time delays, show really a substantial amount of signal. And a trend that we noticed, which is kind of shown here, and you'll see this in several other figures that I'm going to show, is that the electron density, the difference density, is strongest at the shortest time delay at 20 nanoseconds, and then sort of dissipates by 20 microseconds, and then gets a little bit stronger as we get out to 200 microseconds. So that's kind of the trend that we've seen. So the first thing I'll talk about is the shortest pump probe time delay, 20 nanoseconds. And we started looking at these maps, and we noticed that they have a really kind of consistent feature across the entire protein molecule, which is that the atoms tend to have negative peaks on them, and those negative peaks tend to be surrounded by positive halos. And you can see that it's very pronounced for these sulfur atoms, which of course are the heaviest atoms in the lysozyme molecule. And what we hypothesized is that these density features are actually resulting from an increase in a B factor. So if we think about the B factor as representing kind of a simple Gaussian distribution of electron density about some mean position, here this plot just shows some toy examples, right, of distributions that have that are sort of broader and broader. And you can imagine that if you were to subtract this red Gaussian from the blue one or the green one, what you would end up with is a curve that has sort of positive features at the far negative and far positive regions, and then dipped down into the negative around 0 here, right? And this is essentially what we're seeing in 3 dimensions, negative peaks on top of our atoms surrounded by positive halos. So we thought, this really seems like it's representing an increase in B factors, an increase in vibrational motion. And so to test that hypothesis, we made some simulated maps where we basically took the dark state structure factors and we multiplied their B factors by 1.2. So we increased the B factors by 20%. And then we made a simulated difference map where we took these structure factors with inflated B factors and subtracted the structure factors with the original B factors and used that difference to generate a difference map. And what you're looking at now on the bottom is that simulated difference map with the artificially inflated B factors compared to the observed experimental map at 20 nanoseconds on top. And I think, and I hope you will agree, that the simulated map and the calculated experimental map have really quite similar features. And if we calculate a real space correlation coefficient between the simulated and experimental maps, we get a correlation coefficient of almost 0.7 across the entire unit cell, which I think is quite good. So I feel somewhat confident saying that we think we have figured out that what's going on here, right, at these very short pump probe time delays, what we see in the difference electron density is evidence for increased vibrational motion. We can also model more specific excited state conformations. So over here on the left, this is a difference map that was collected for a 20 nanosecond pump probe time delay. And you can see very clearly density for this tyrosine residue that represents rotation of this rotamer. And that's exactly the type of conformational change that you would expect to see on sort of the nanosecond timescale, right? Rotamer exchange is frequently associated with those types of timescales. And then at longer pump probe time delays, like 200 microseconds, shown here on the right, we see features in the map that correspond to larger amplitude motions. So here you see a loop region corresponding to about 4 amino acids. And we're only showing the backbone carbon, oxygen, and nitrogen atoms here to kind of simplify the visualization. But you can see quite clearly that the structure moves from these negative peaks. The ground state structures in each of these figures are shown in gray. The structure appears to move right from these regions of negative density into the regions of positive density. And so the dark green models represent our manual model based on the electron density. And then the light green models show what results from extrapolated structure factor refinement of the dark green models. So what that suggests is that we're able to model excited state conformations that are present at relatively low populations, and those refinements are sort of stable, and the excited state conformations that we've modeled don't really collapse back to the ground state. So we feel like the signatures in the electron density that we're seeing for these alternative conformations are real. We also wanted to ask the question of whether or not the motions that we're seeing are actually functional, or if they're just kind of a byproduct of heating the protein up, right? Because anytime you heat up a molecule, it's going to move around more. And so we want to make sure that what we're seeing is not just random motion, but actually the functional movements of the protein. So to start to address this question, we used a ligand called chitobiose. And chitobiose binds to the active site of lysozyme. You can see it in the active site cleft here. And it basically locks the enzyme into the closed conformation. And so we repeated our temperature jump measurements using crystals where we bound the protein to chitobiose. And what we noticed is that the binding of this ligand to the active site substantially changes the evolution of the time-resolved signals. So what I'm showing on the top of this slide are images of electron density for that region. Let me go back. For this loop, comprising residues 97 to 100. So you're seeing that region here. And in the top panels, you're looking at the electron density as a function of pump probe time delay for the APO enzyme, so no chitobiose. And just to kind of summarize what I've already told you, we see these very strong features over the atoms at 20 nanoseconds, which we think correspond to increased vibrational motion. As we get to 20 microseconds, that density kind of dissipates. And then at longer pump probe time delays, like 200 microseconds, then we start to see evidence for concerted shifts of loop regions, such as the residues from 97 to 100. So this is what we see for the APO enzyme. For the chitobiose bound samples, we see something quite different, which is that at very short pump probe time delays, 20 nanoseconds, the electron density features look almost identical between the APO and the chitobiose bound structures. You can see this valine residue, for example, has peaks out at the 2 gamma carbons. You can see quite a few peaks on backbone carbonyl atoms. But in contrast to what we saw for the APO enzyme, where the signals change substantially by 200 microseconds, in the case of the chitobiose bound enzyme, we don't really see much of a change in the characteristics of the electron density as we go from 20 nanoseconds to 200 microseconds. So what we think is going on here is sort of summarized in this last slide that I'm going to use to talk about our lysozyme work, kind of describe the model that we've put together here. So we think what we're seeing in our T-jump measurements is the early events of this clamshell motion that I've been describing throughout the talk. So in the laser off state, we have our sort of resting APO configuration. And when we apply the temperature jump, we get a rapid increase in vibrational motion. That's what these little red dots are supposed to represent. And then as we get to longer timescales out to the microsecond timescale, in the APO enzyme, those vibrational motions dissipate into larger scale motions, like the motions of this loop. And I have the structure of the actual protein here to show that this loop that we're talking about, this 97 to 100 loop that we see moving, is right above the active site, right? So we think that what we're seeing here is sort of the early steps in the closure of this enzyme active site. And in contrast, when we bind chitobiose to the enzyme, chitobiose locks the enzyme in the closed conformation, even in the absence of any temperature jump being applied. And so then when we hit this with the laser and we increase the vibrational motion in the enzyme, it's already in the closed conformation. So there's nowhere for it to move to. So rather than those vibrational motions dissipating into these larger amplitude loop motions, the enzyme just stays locked in that closed configuration, and those vibrational motions persist out to longer timescales. So this is sort of the model that we've put together for what's going on based on our early experiments with lysozyme. And again, we have a very limited set of pump probe time delays here. But what was important for us in this work on lysozyme was to kind of demonstrate proof of principle and show that we can do temperature jump experiments with protein crystals and actually observe changes in the electron density that were meaningful. So at this point, I think we had sort of satisfied that goal. But of course, lysozyme is not a very exciting protein. And Sakura, Japan, right, is very far away from California. So we pivoted from doing these experiments on lysozyme. And when I started my own group, we wanted to expand the temperature jump approach to other proteins, right? Real proteins that have actual interesting biological questions that we can ask, not just model systems like lysozyme. And also at UC Merced, we're fortunate to be about 2 hours driving distance from the only free electron laser in the United States. So it seemed kind of ridiculous to travel all the way to Japan to do these experiments when we have a free electron laser essentially in our backyard. So relatively early on after I started my group in 2020, we struck up this collaboration with folks at LCLS, as well as Mark Wilson, Alec Fulmer, Ryan Haight, and Sergio Carbajo, to develop a standard configuration for doing temperature jump experiments at LCLS. And so I don't want to go too much into the details about this here because I'm already sort of running out of time, but this setup involves a nanosecond OPO similar to what we have for the IR experiments that we did at SACLA. That OPO is delivered to the interaction region using a similar sort of free space configuration. We also deliver the sample using a similar high viscosity extrusion approach, like what I showed for our lysozyme crystals several slides back. And really kind of the key takeaway that I want everybody to take away from this slide is that we can now do temperature jump experiments at LCLS at the MFX end station. And this standard configuration is available broadly to users. So if you're interested in doing temperature jump crystallography at LCLS, you can contact Mark Hunter. I also will pause to say that there has definitely been enthusiasm about temperature jump crystallography at Biocars. So I encourage you to also talk to the Biocars folks about the potential to do experiments there. And yeah, this is sort of, you know, I like to think that it's becoming mainstream, the ability to do these temperature jump experiments. There may not be a lot of people doing them yet, but the ability to do them is now sort of available to users. This is just a short video showing how the experiment runs at LCLS. We have a similar high viscosity extruder to the one that I showed for our experiments at SACLA. Here, this video is not playing. Seems like my video is not playing. I was just going to show the experiment running. It's not that important. And once we developed this new configuration at LCLS, we wanted to apply it to an interesting biological question. And so we sort of stumbled upon the enzyme soybean lipoxygenase. And soybean lipoxygenase is quite different from lysozyme. It's a 95 kilodalton enzyme. So it's almost 6 times the size of lysozyme. And it's a non-heme iron enzyme. So this little purple atom at the center of the SLO structure represents an iron atom that is not bound to heme, but is involved in the catalytic mechanism of SLO. SLO catalyzes lipid hydroperoxidation. It's a relatively complicated reaction scheme that's shown here. The enzyme starts in the iron 3 hydroxide resting state. The lipid substrate, which is annotated RH, comes in. The first step of this reaction involves proton-coupled electron transfer that transfers a hydrogen from the substrate onto the iron center and leaves a lipid radical in the active site. Then there's a second step that involves oxygen insertion into the lipid radical, and then a rearrangement of the iron center back to the iron 3 OH resting state. So it's a relatively complicated reaction. And the reason that we started working on this enzyme is because I was approached by Judith Klinman, who has actually been working on SLO for decades. And what Judith's work has demonstrated is that the rate limiting step of this reaction is this first PCET step. And she's shown that this PCDT step, this rate limiting step, proceeds via hydrogen tunneling. And that was demonstrated based on an extremely large kinetic isotope effect of nearly 80 at room temperature, which is huge for an enzyme and implies the existence of tunneling in this mechanism. But interestingly, and down here, right, you can see some results from her early papers showing that the catalytic rate for the proteated, or the proton containing substrate is much higher than for the deuterium containing substrate, right? So this is the KIE of approximately 80. And interestingly, she also showed that this KIE is temperature dependent. And so for a purely quantum mechanical phenomenon like tunneling, you would expect the KIE to be temperature independent. So the fact that Judith observed this temperature dependence suggests that the tunneling event is somehow coupled to the environment, and in particular to the temperature dependent dynamics of the environment. And so this is an idea that Judith has been pursuing really since the 1990s. I'm going to skip that figure. And really, her most recent paper kind of very elegantly describes her hypothesis. So what she believes is going on in this enzyme is that the substrate binds to the metal center, and the enzyme undergoes some conformational exchange, and certain conformations of the enzyme are capable of funneling thermal energy from the solvent to the active site. And that sort of thermal motion through the protein scaffold basically shortens the donor acceptor distance, right? So the donor in the tunneling reaction is the C11 position of this lipid. The acceptor is the iron OH species in the active site. And so her hypothesis is that the dynamics of this enzyme, the thermal dynamics of this enzyme, right, are responsible for shortening the donor acceptor distance and facilitating the tunneling reaction. So she approached me and said, do you think we could use temperature jump approaches to study SLO and to visualize the motions that are responsible for tunneling? And I said, absolutely, we could try to do that, because that sounds like a really cool biological question that's much more interesting than lysozyme. So we grew some SLO microcrystals, we mixed them into cellulose, much like we did for lysozyme. We can collect some very nice X-ray diffraction patterns from our SLO crystals. And when we started processing the diffraction data, we observed that these crystals are really, really polymorphic. So what you are looking at here, let's focus, I think, on the histograms in the middle. You're looking at the distribution of unit cell axis lengths for the A, B, and C axes for the thousands of crystals in this data set. Down here on the bottom are similar histograms for the alpha, beta, and gamma angles. And these are really heterogeneous, right? In an ideal serial crystallography experiment, you have really nice sharp distributions here that suggest that all of your crystals are isomorphous and they can all be merged together. But we saw this and we were really quite intimidated. So we thought maybe this is a batch like a batch to batch variation problem. So what we did was we plotted the same distributions of the A, B, and C axes as a function of basically run number. So progress through the experiment is one way to look at this. And we realized that we really have two groups here. One very highly polymorphic group, and another group that is still polymorphic, but a little bit better. So we decided to pursue the less polymorphic sample. So we went ahead with just the portion of the experiment that's represented inside the green box here. And we now made some plots showing these clustering plots, where instead of just showing histograms for a single axis length, now we're showing the both the A and B axes on these plots, right? This is sort of a two-dimensional plot. And you can see these two clusters, the red cluster and the blue cluster. And what we noticed by also plotting the unit cell volumes for the crystals in each of these clusters, we noticed that the volumes are actually the same. So probably what's going on here is that the red cluster and the blue cluster both actually correspond to the same crystal form. There's just some kind of indexing ambiguity. And so what we realized is that SLO is actually a really, or at least the crystal form that we had been using, is a really bad crystal form for serial crystallography, because not only is it in P21 space group, which is polar, and so you have this polarity ambiguity that's inherent to merging the serial data, but the A and B axes are almost identical in length. So what this results in, and I'm running out of time, so I'm not going to go through all the details, but there's basically one correct way to index your SLO crystals, and there are three incorrect ways to index your SLO crystals. One where you get the A and B axes swapped, one where you point the C axis in the wrong direction, and a third where you've screwed both of these things at the same time. So we needed to basically figure out how to re-index all of the incorrectly indexed images and get them to match to the correctly indexed ones. And so the way we decided to do this was basically to take each of these clusters, process the data in P1. We take one of the clusters then and swap the A and B axes so that the A and B axes match. Then we move from, for both clusters, we move from P1 to P21. So we actually invoke the symmetry now at this point. And then at that point, we can resolve the orientation of the C axis using the sort of classic Bremen-Diedrichs type of approach that's used to solve indexing ambiguities for polar space groups in serial crystallography. So we were able to solve this problem, and this is some heroic work from Alex Wolf, as well as Aaron Brewster and Dan Paley. So we developed this method, and then with that in mind, we kind of went back to the more highly polymorphic data shown in the red box here, and realized that we can actually sort this data out using our clustering algorithm. And I'm gonna save everyone the details again in the interest of time, but basically what we realized here is that in this more this highly polymorphic crystal batch, we can basically solve two different structures, one corresponding to a small unit cell, right, with a lower volume, and one corresponding to a larger unit cell with a larger volume. And those different structures are actually slightly different. So what I'm showing here is the SLO molecule in the small cell on the left and the large cell on the right. And this is anisotropic ellipsoids that are the result of a TLS refinement. And you can see that there is a lot more interdomain motion in the large unit cell than in the small unit cell. So over here on the right side of the protein structure is this little beta barrel domain. And you can see in the large unit cell, the anisotropic ellipsoids are much larger, right? So this larger unit cell sort of accommodates the motion of these two domains a little bit more than the small unit cells. So whether or not that has relevance on the T-jump induced dynamics, we're still not sure. But I wanted to point out kind of an interesting feature of these data sets that we can actually, for better or worse, solve two different structures from the same batch of crystals with a little bit of careful data processing. Of course, we want to ask whether or not we can see temperature-induced dynamics. So what you're looking at here, I think we can, for now, ignore the one-dimensional plot at the top, and let's just focus on the protein structure. You're looking at the structure of SLO with its C-alpha atoms shown as spheres, and those spheres are colored according to the amount of difference density that is within 3 angstroms of that particular residue. So this is just a very rough way of assessing whether or not we observe T-jump induced structural changes based on difference of electron density there. And the coloring goes basically from white to red. The more white a sphere is, the less density we see near that amino acid. The more red, the more density we see. And this black sphere in the center, just for reference, is the iron atom. And on this slide, I'm showing the control measurement for SLO. So this is a dark minus dark difference map. And so the amount of difference density that we see as represented by the colored spheres in this image shows sort of the noise level of our SLO measurement. And now I transition to our experimental measurement where we're subtracting the laser off data from our T-jump data. And again, coloring the spheres according to difference density in the vicinity. And you can see that there's this one side of the protein where we see a large enrichment of features above what we see in the control. And this is really interesting because over many years, Judith has identified different regions of the protein that she believes is involved in the dynamics, these dynamics that drive catalysis, that drive tunneling in the active site. She has used techniques that lack atomic resolution, like hydrogen deuterium exchange, mass spec, or she puts fluorescent probes on different parts of the protein and measures Stokes shifts. So she does all of these biophysical experiments that lack atomic resolution. And the different regions of the protein that she's identified as being important are the ones that I've colored in blue, green, and pink. And so the regions where we see the most difference density kind of ties right in with the part of the protein where Judith has already identified important dynamics. So we're excited about that. I'm going to skip that slide. Also, here I'm showing a similar depiction of the protein, but instead of the C alpha atoms shown as spheres, I'm showing the ordered solvent molecules as spheres, and again, coloring them according to difference density. And you can see that there's a lot of difference density changes around the solvent molecules as well. And this is also consistent with Judith's hypothesis that these motions within the protein are driven by collisions with the solvent. So this, the data that I'm showing here corresponds only to a single pump probe time delay, which is 100 nanoseconds. More recently, we had a beam time where we were able to sample 5 more pump probe time delays. We got 0, 16, 29, 63, and 100 nanoseconds. So now we have 6 different pump probe time delays, and we're in the process of working through all of that data right now, but we hope we can bring more kinetic detail to the data that I'm showing here on this slide. We can see just from a single pump probe time delay that there are some interesting temperature-induced changes in this protein structure, and we're hoping that with this additional data that we collected recently, we'll actually be able to map the time-resolved changes that are sort of at the heart of this. I know I've already gone a couple minutes over, but I just want to say one last thing because I think it's really interesting. So one other benefit of doing these experiments at the free electron laser is that you get data sets which are ostensibly radiation damage free, right? So these data were all collected with 30 femtosecond or less X-ray pulses. And so, as I mentioned before, right, we get the diffraction data before the onset of any radiation damage. And so this allows us to look really carefully at the coordination geometry around the iron in the SLO active site, which is something that's never really been done before, mainly for reasons related to radiation damage at the synchrotron. And so in addition to all the data that I showed so far are actually for the reduced iron 2 state of SLO. It turns out that when you purify the protein from E. coli, it just comes out in the reduced iron 2 state. So we work primarily with that. But it's relatively straightforward also to oxidize to the iron 3 state. So this data has no temperature jump at all, but basically just shows the radiation damage-free view of the iron coordination in the active site. And what you can see, and what we believe we're seeing for the first time in a crystallography experiment, is that the ferrous iron 2 form of the enzyme is a 5-coordinate iron center with the key OH group right here. And then there's the C terminus of the protein and 3 histidines form the rest of the coordination sphere. And then when the enzyme gets oxidized to the iron 3 state, this key hydroxide moves over, and this oxygen from the asparagine moves into a much shorter distance so that now it's actually part of the coordination sphere. And now we have a nice 6-coordinate square planar coordination geometry. So This has been studied using spectroscopic methods, but this is the first time we're actually seeing it in crystallography. And the reason that we think this is important, perhaps relevant to the dynamics, is because it's an outstanding question about whether or not SLO uses this, what I'm referring to here as an oxygen preloading strategy. So basically what that means is that the oxygen, this O2, actually binds to the enzyme before the reaction cycle is initiated. And that's because once you form this lipid radical in the active site, this species is very reactive. So if this oxygen isn't already in the active site and ready to go, then this lipid radical can perform other unfavorable side reactions, right? That's not what you want happening in the active site. So there's evidence for this preloading of oxygen, and in the iron 3 structure, oops, sorry.
Oops.
In the iron 3 structure, we see, wow. Oh my gosh.
Okay.
Anyway, you know what? That's basically the end of my talk. So I'm just going to finish with words and no slides. In the iron 3 structure, we see a little bit of extra electron density, which we can attribute to oxygen speculatively. And so we think that not only do we see this shift in the coordination sphere, but we also see the binding of oxygen. So that's all of the data that I had to share. And I already did my acknowledgements at the beginning. This is one of the reasons why I do that. So I will end there and sort of thank you for your attention. And I'll try to get my slides back up while people think if they have any questions.
Okay, thank you so much, Mike. It was a wonderful talk, very, very interesting results and very relevant to what we do at Biocars. Perhaps we are running late, but perhaps we have a couple of questions. We have time for a couple of questions if anybody would like to ask. Just unmute yourself, introduce yourself, and ask a question if you have one.
I have a question. So this is Yulia Pushkar, Purdue University. So I have two questions. One is technical, and it relates to very early part of the talk, where the temperature was reported with 10 Kelvin precision. So I'm wondering how the temperature was controlled, because sometimes people using like cryostreams. So that's a technical question. And then there will be one more fundamental.
Yeah, so I'll answer that question first. So unfortunately, we don't have really tight control over the temperature when we do these experiments at the free electrons. So
no, so that was your first nature chemistry on the crystals, the very beginning of your talk.
Oh, that was solution.
So those experiments were done at Biocars, the solution scattering work, and that was a collaboration with Philip's group, and they
sorry, they were crystallographic data, so they would like density maps on the right, and like 300 Kelvin, 310 Kelvin.
Yes, those experiments. That was done using a cryostream, yes.
But how you can control with the cryostream to the 10 Kelvin precision?
Oh, the cryostream has very high precision. I mean, within, tenth of a Kelvin. And we've actually measured this using not only scattering methods, but also just by putting like a small thermocouple on the end of our goniometer pins.
Okay. That's great to know. Okay, and then
stored beam has been lost. We're investigating. Stored beam has been lost. We're investigating.
Okay, lysozyme work with the molecule bound in. So fundamentally, you would think if you do the temperature jump, you will move to higher energy conformation. And you kind of showed that in one case, you're kind of moving towards closing, which is a high energy conformation. But in the second example, you showed that basically temperature jump makes no difference. And this is kind of contradictory. Basically, in that case, maybe it was supposed to open in because, you increase the energy in the system. You need to move to high energy conformation. So I want to understand more about that.
I think inhibitor binding sort of changes the depths of the wells, right? So binding to the inhibitor makes the closed conformation basically the only thing that can be populated, right? And so now the open conformation is at such higher energy because in order to get there, you have to pull it open and release the inhibitor. And so now you're just shifting everything into that closed conformation, even though in the absence of the inhibitor, that's the conformation that's at higher. So
but then it, yeah, I got it. But then you have to kind of a rule or something. Do you have like any way of doing computation and predicting those energy scales and then saying, okay, for those kind of conformations, T-jump will be effective, but those are too high off for T-jump to kind of move a needle. So that would be great to know the energy scales.
Yeah, I agree. That's actually a really good idea. And we had never thought about that before, but that's a really good idea. And we have sort of, I would say we've moved away from lysozyme a little bit, but this is a wonderful idea that I'm going to keep in mind for, some of the other systems that we study.
Okay. Okay, now Arnold, it's your time to ask a question.
Yeah, thanks. Yeah, my name is Arnold. I'm a student in Lin Chen's group at Northwestern. I have two questions. One is, I'm not very familiar with time-resolved X-ray crystallography, but I know in solution scattering, in T-jump experiments in particularly, we can sort of change the accessible conformations by changing that background temperature, right? That initial temperature. Is there something analogous that you can do in X-ray crystallography in which you can change like the accessible conformations, because oftentimes the laser T-jump is fixed?
Right. Yeah. So that is, in principle, you could do the same thing. But as sort of a technical comment, I'll say that the work that we've done has been almost entirely at free electron lasers. And they are not well designed to control the sample temperature, at least not in the configurations that we use. So we are sort of always kind of stuck from jumping from like ambient temperature up to wherever we can get with the laser, right? So we've been sort of limited so far. A major, and my PowerPoint crashed, so I didn't get to put up my conclusion slides. But one of the things that my group is really interested in moving forward is continuing to develop the hardware and the software for doing these experiments. And so we've been talking a lot with the folks at LCLS about how we could get actually proper temperature control for these experiments so that we can do exactly the same type of thing that you're describing, right? You can change the starting temperature, and then that enables not only to kind of control the distribution of conformations, but also to do things like, proper ironing analysis, and then you can do, calculate like transition state thermodynamics and things like that. So there's a lot of really good reasons to want that level of temperature control, but unfortunately we don't have it. That's one thing where doing temperature jump crystallography at biocars, I think could have advantages over the free electron lasers. Biocars has very, good systems for controlling sample temperature. So I think that there's probably a lot of opportunities.
Gotcha. Thank you very much. If there's still time, I have a follow-up. Or not a follow-up, but I have another question. It's more about the science of studying enzymes with crystallography. Were there any attempts to look at co-crystals, or are there plans to look at co-crystals with a substrate? And, how might that affect some of the dynamics you see after T-jump?
So absolutely, it could affect the dynamics. And we would love to do that. Unfortunately, so there's a couple of problems. The biggest problem for us and with our current work on soybean lipoxygenase is that it doesn't really crystallize with the substrate bound, which is kind of an interesting problem, right? Like clearly the substrate is changing something profound about the protein that makes it hard to crystallize. But the work where people have tried to crystallize the substrate with the enzyme, you have to like mutate the active site residues, for example, right, to make it so that it no longer reacts. Another big field in serial crystallography is doing like mixing types of experiments with enzymes where you can actually mix in the substrate, right, and then capture like a bona fide enzyme substrate complex. So we have started, and this is in collaboration with Mark Wilson, we've started to do, this is not on soybean lipoxygenase, on a different enzyme, but we've started to mix substrates into crystals, form the enzyme substrate complex, and then do a temperature jump on it. Because with the mixing experiments, your timescales are much slower, right? So you can capture enzyme substrate complexes that are stable for milliseconds. But with the laser temperature jump approaches, you can look at dynamics that are sort of nanosecond to microsecond. So our strategy has been to form an enzyme substrate complex that's stable for milliseconds. And during those milliseconds, we T-jump it. And so we're kind of moving in that direction, but it's very early days.
Yeah, sounds great. Thanks so much for answering those questions.
I know we're running quite late, but do I have time to maybe make a few comments, Michael?
Okay,
Sure don't,
okay.
you don't mind, and it helped that we're late, so please do. I'll stay.
So
Okay.
one thing I would encourage is with your extrusion nozzle, it would be nice to have temperature-dependent control on that. And then here's why. You showed how when you do a T-jump of nominally 10 degrees, that you increase the B factors. And now you're comparing light with dark. And a lot of the differences you see arise from the differences in the B factors. Now imagine if you could collect dark data at the elevated temperature that you achieve when you do the T-jump. Now the B factors are going to be the same. And when you take differences, you will be able to visualize the intrinsic protein contribution to those differences. So you will be out of equilibrium. And now when you track as a function of time, what you would see is those differences would gradually disappear on the time scale at which you equilibrate and you come to equilibrium. So that would help you focus on the early dynamics in a much clearer way, I believe. But that would require being able to control the temperature of that extrusion nozzle in a reasonable way so you could match the T-jump magnitude that you can measure. So this should all be doable, but I think it would make these small differences that you see easier to extract from the data.
That's a really great idea, and yet another reason that we need to get the temperature control on that nozzle. It's been, it's been a source of consternation for me for quite some time, but we haven't gotten a good solution.
Yeah. Then another point regarding that, when you do a T-jump, you generate a very far out of equilibrium situation. So when you jump the temperature that quickly, it's an isochoric T-jump. There's a constant volume T-jump. But because of that, it's out of equilibrium, it has pressure. So the pressure goes up dramatically. And that causes a volume expansion. And that changes the density of the protein in that path. And the thing is, as it expands in response to that, it actually goes to negative pressure. And then it will come back and oscillate. So there's going to be a photo-generated acoustic wave that will modulate the pressure on. And there are people like Saul Gruner who have shown pressure-dependent changes in crystal. And so, you would expect to see some differences there. So when you look primarily just at, say, 20 nanoseconds, that's at the height of the pressure. But what could be also interesting is it requires higher density time points. And the oscillation period will depend on your sample dimension. So when you say 75 microns, my guess is that would be of the order of 50 nanoseconds. It's just a very rough guess that you might see oscillations on that time scale. And so what you would want to do is acquire time points dense enough, kind of linearly, in that early time frame. And then you would be able to also capture what differences arise from pressure differences. What differences in structure can arise from pressure differences? Of course, that will damp out by the time you get to a microsecond, that should be damped out. But the magnitude of that effect might not be small. So when you measure just selective times early on, you could be looking where you have the high pressure, or you could be looking where you have the negative pressure. And that could lead to quite different interpretations of what you've got. So again, I would encourage you to look at these early, when you're using such short excitation, to look at dense time points early on and see if you can see those oscillations, because you would expect more dramatic changes during that early time window.
That's a great point. I remember doing experiments with you and your group and seeing these oscillations in the data.